mechanics, biological physics, and he's participating in the school going on that Roberto's organizing, so he'll give 2 lectures at the school, and then he'll be around next week for the workshop. So he'll be around, I guess, for 10 or 11 days, so people can feel free to talk to him. So today he's going to talk about generating transition paths with Markov bridges and its application to self-hate choice. Thanks.
Is it working now? Okay, all right, sorry. Yeah, so I'm coming from San Francisco at the Zuckerberg Biohub. And today I wanted to talk about some of our recent work. And it's a method to generate transition path. And so I'll talk about it for most of the talk. And then, because we're in an institute with a lot of application in biomedicine, we applied it to a case to study self-hate. And so I'll run through this also during the talk. I'm into this theory group, which is run by Greg Huber at the Zuckerberg Biohub. We're a small group. And so I'm just putting this here to thank them for their help during this project, and also some of the people who helped me to understand those big data sets of self-hate. Okay, so this is an outline of what I wanted to talk about today. I'll start with some motivation, then I'll go through the method and what exactly Markov bridges are, and then I'll go through the application to investigate self-hate choices. Okay. Well, what's a textbook view of transitions? I think most of you have probably this picture in mind, where you are, when you want to study transition, for example, in a chemical context, it's very common to represent, one given state, which could be the reactant state, and then the final state, which could be the product state, B. And what's happening is that during the reactions, so this could be a reaction coordinates, you're going through a sequence of states which have different energies. So this is the energy axis. And those states make you go through a state which has a very high energy. And this is what I call the energy barrier, right? And since the 20th century, we know some things about it. For example, the Swedish physicist Arrhenius, he proposed an empirical law at the time, which was that the kinetical rate in this setting to go from A to B should be proportional to the exponential of minus the height of the energy barrier. And this was later refined in the 20th century. The Dutch physicist Hans Kramers, he showed from first principles that this was actually true. And in fact, if you take the inverse of this relationship, 1/k is the typical times it takes to go from A to B, and this is called the Kramers time. And this is exponential into plus the energy height, right? So this is the time scale we're talking about.
Sorry, what is R? R is just a parameter?
R is the Joule constant. So RT equal KBT, yeah. And so why am I even talking about this? Well, in a lot of settings, it's interesting to understand transitions and especially the path you're taking to go from one point into another. So this is a very abstract representation of what I'm saying. You could, in this case, it's a two-dimensional plot. You could see them as reaction coordinates or maybe space coordinates. And the question you generally ask is, you have like an initial state A and a final state B, and you want to know how you go from A to B, and maybe some paths are more likely than others. But you will also want to know, what are other choices and what are their relative statistical weights. This is a question which appears in many different fields. For example, in protein folding, the general question is, this function here would represent the potential energy landscape of the protein, So here it's 2D, but in general, it depends on all the coordinates of the monomers of the protein. But if you do maybe, for example, you isolate some reaction coordinates in 2D, you would have something like this, where you have a very rough energy landscape. And in there, on the edge is usually the state of the protein when it's unfolded, right after translation. And the native state is the minimum of these functions, but to go through there, you have to go through a very rough landscape, and you could be stuck in several energy minima. In general, this is a tough nut to crack, and there are different ways to get there, and there are different ways to get stuck in intermediate states. Okay, this is a little simulation of what it could mean to study protein folding with molecular dynamics, for example. Another area, for example, is this case, which is called the genetic toggle switch. It's one of the classical examples of a system which is bistable. It has two stable states, A and B. And though this here, if I explain it a little bit, it's like suppose you have two proteins, A and B, which can be produced by the same operator. So an operator is like the promoter on the DNA, right? And the thing is, A can dimerize, and when it binds to the promoter, it represses B. So the states when A is expressed, blocking B means that the majority of, you're going to have a lot of A molecules around, and this is one stable state. But because of leaky expression, you could start inversing the trend, and B could start, agglomerating and binding this promoter, and then you can have a transition into the other symmetric stable state, which is B. And so if you look at here, the two coordinates would be the number of A molecules and B molecules. You're going to have two stable states, a lot of A, very little B, and a lot of B and very little A. And you're going to be able to transition between those two states. And how do you transition? What are the trajectories and what are their relative weights? So here's an example of a paper like where they showed several possible trajectories, and the majority fall into this line here. Another example of application of how cool it is to understand those transitions is maybe in a more applied mathematics context, where I think you all know about Brownian motion, right? And so I'm showing here in 1D the 1D Brownian trajectories of a particle diffusing. You have the time here, and then the y-axis is the position. And bridges in this context would be a particle which starts at 0, diffuses, and then go back at 0. And if you do this little trick where you identify the minimum of these trajectories, and then you patch the curves, like you move the red one to the left and the blue one to the right, you get these constructions, which is called an excursion. This is harder to generate, without tricks. And so understanding transitions like this in this context allows you to generate excursions, which are notoriously difficult to generate. So this is just to try to say why, in a wide variety of contexts, it is useful to understand transitions and to know how to generate them. Okay, what do we have already? What is known, and what are the methods that you could pull out of your quiver? I'm dividing them into two classes. Classes which first applies to the continuum. And so what do I mean by that is you typically want to consider systems in which, you have like a variable, a vector variable, which follow a Langevin dynamics type of equation, so it's multidimensionals. And the Langevin dynamics equation is a good equation to study thermally activated processes. So this is the derivative of the position. This is the force. So this is the the gradient of the potential U is the force. To the sign, and then the eta here represent some noise, so white noise. You may or you may not know that you can't formally solve this equation, and for example, the probability to be in a given state, in the final state, given the initial state, this can be written as the exponential of the difference between the energy of the two states, and then an integral, which is a path integral, and this presents some analogy with quantum mechanics, but I'll come back to this in a moment. But so in this path integral, all from there, you have like the you're integrating over all the possible paths between the initial and final states. And then in the exponential here, you have, this is an action, and the action are integral over these terms, which contain some, what in quantum mechanics would be the inertial, the velocity term, and this would be the effective potential. And here, the effective potential is related to the definition I put there through this transformation. So this is known, and why am I talking about this? Well, because you can leverage this representation to understand those transition paths. So for example, because you have this representation, you can figure out what is the optimal path through list action principles, right? So you have integrating over all possible trajectories of the exponential of an action, and so you know that the optimal path would satisfy, you can find it by using the action, which must satisfy Euler-Lagrange equations.
Sorry, what happened to eta?
Sorry?
Why didn't eta enter into the probability? So it looks like eta equals 0. I'm asking if the probability that you defined depends on eta.
Depends on what? The stochastic. Oh, eta, yeah. But well, eta is, yeah, of course. Eta here is usually, the variance of this white noise is going to be equal to 2D in general applications, 2 times the diffusion coefficient. But this is the Langevin representation, right? So then the usual, how you go to there is you transform this into a Fokker-Planck equation.
I'm happy to go if, I'm just asking, is that probability when eta equals 0? It looks familiar to me, that probability. Is that for eta equals 0?
Which
is that when the variance equals 0?
No, this is the, is in general.
I'm sorry, why doesn't that depend on eta? I don't see eta appearing in the probability.
8? Eta. Sorry. Well, I'm not sure.
You can use the cloth to erase if you want.
The cloth?
There's a cloth right there.
Yeah. So there is this Langevin equation that I wrote, right?
Yes.
And so this thing here is, it's a white noise, so it has this, right? Yeah, D.
Okay, thanks.
Thanks, sorry. Okay. Yes, so so optimal path. So then you have you can get the optimal path by solving the Euler equation associated with that. There are other methods which have been proposed in the last 20 years or so. You have what's called dominant folding pathways, and so those are methods which try to, from the solution here, generate trajectories which are close, statistically from the dominant one. And more recently, you've had the emergence of this Langevin bridges methods. And here, a bridge is a path which satisfies, a Langevin dynamic equation initially, but which is conditioned to end in the final state at xf at time tf. And so those methods are, statistically exact, but the conditioning enters the equation through this additional term. So you have like a potential Q, which depends, of course, on all the parameters here in these variables, but which incorporate this condition into the final state. And you can show that basically the paths generated by this method preserve the statistics. And so you would get the same path if you were doing these methods and just filtering those that finish in the given end states. And if you look at those statistics, it will be the same as this one. But those have become very useful in different areas of physics. So these are examples of to super diffusion. I've never seen it, so I'm not sure. So these are examples of Langevin bridges that you would get. So this is a landscape you'll see later, so I'm not going to pay too much attention to it, but you would have in white the dominant path or the optimal path, and then in the colored one corresponds to realization of those Langevin bridges. So they all start from a well labeled A and in a well labeled C, and most of them are around this dominant path, but you have some rare possible paths which actually go to some convoluted pathways, which are very unlikely. Okay, so this was for the continuum case, but now I'm going to talk about what's available for discrete systems. So what I mean by discrete systems is a system which satisfy this type of master equation. So they are so-called discrete Markov jump processes. So in this equation, P alpha of t would be the probability to be in state alpha at time t, and the W's represent rates. So this one here is the rate to go from state beta to alpha in a small time interval. So how would you answer the same question? Well, you could do it by brute force. You could use kinetic Monte Carlo to sample realization of those paths and then filter those that end up in the state that interest you, and then look at the statistics. You have different methods available. You have the Gillespie algorithm, for example, or the next reaction method. This is possible, but as you might expect, it's very inefficient. Other methods which are approaching this question are, for example, this forward flux sampling method, which uses some sort of a ratcheting scheme to answer the question. And so starting from a certain state A, the method defines several interfaces, which it's a sequence of interface in phase space, which then goes from A to B. And then you're going to run simulations from A to the first one, and then you stop. And then you're going to run simulations which goes from this lambda 1 to the lambda 2, and then you stop. And then you do this until you reach B, and then you patch them together, and because of the Markov properties, you have some trajectories, which are transitions. Okay, but realistically, to my knowledge of this, that was mostly it. And so the rest is going to be the topic of this talk, and so I'm going to focus on those stochastic Markov jump processes and how to have the equivalent of Langevin bridges. Here the jumps are not Gaussian. I mean, they could be Levy, I suppose, if your rate coefficients have strange distributions. Yeah. So everything is encoded in the matrix of rates. Yeah. Okay. So brief words before I move on to the core of the talk is this is a very simple example of a particle diffusing in the double wave potential, and so. This is the movie, but if you were looking at the trajectories, what you would see that the position of the particle goes between -1 and +1, which are the two wells, and this is time. And so what's happening is it spends a lot of time, dwelling in a specific well, and then it makes a transition. It spends a lot of time dwelling in the other well, and then makes a transition. And what you get from there is that the time it spends transitioning is actually much shorter than the time it usually spends dwelling or sampling a given well. And so you have this curse of time scale, because if you want to do these transitions with the method I just shown, in general, you also have to sample this time, which is a long time scale. And so, like if I was starting the simulation here, I would have to wait all this time from there to there before the transition happened, while the actual time scale of interest here is very small. So, yes. Yeah, I forgot which temperature I chose there, but yeah, otherwise, low temperatures, you would get longer dwelling times, and high temperature, it would be hopping a lot, yeah. Okay, so the rare events, like the transitions, are hard to see, and so that's why answering those questions is usually difficult. So back to this slides, I want to add something. There are recent works, and I'll come back to this later, but they've shown that actually what I was just talking about, these times, like you have to wait before you transition to the next state, it's the Kramer time. It's the first passage time. It's the first time you have the first occurrence of the transition into the other well. And this time contains, it contains aborted transition, particles which start to transition but then come back. And so, but once the particle commits to the transition, this is the transition pass time, right? It's really the time it takes to go from the well on the left to the well on the right, for example. And this transition pass time doesn't scale like the Kramer time. I said before, this one scales exponentially the barrier height. This one scales logarithmically in it. So it's much smaller. Okay. And last introduction maybe is how do we sample trajectories like this for these discrete jump processes? Well, you use kinetic Monte Carlo. In a simple diffusive context, I'm assuming a lot of you are familiar with it, but you could just integrate numerically, right? And so you would have like for diffusion with a forward and a backward rate, for example, this would be the probability to go forward in a small interval of time delta t, and then the other probabilities, and you can just iterate like this. There are more efficient methods, like the Gillespie algorithm I mentioned, which are event-driven, so you sample directly the sequence of times where transition to a different state occur. Much more efficient, so this is what I'm using in the rest of my talk. And all of this is very well known, and it's connected to the master equation I was describing in the beginning. It's an exact way to simulate, these equations in the, all right. Okay, so to the core of the problems, how do we sample those bridges? I'm going to start with a case study. And the case study I'm showing here is the potential I was showing before. It's called the Mueller-Brun potential. It's a very famous potential in the chemical physics. It's used to analyze all sorts of activated sampling, to evaluate method to sample activated events. Here the question I'm asking is, the field lines represent the equal energy lines, right? So you have one well in A, one well in B, and one well in C. And the ordering is such that the stable well is A, and then the second most stable is B, and then in between you have C, which is the highest minimum. And if I'm asking to you, how do we go from A to B? Well, you're going to probably start in A, then end up in this minimum, and then go to B, right? And you're going to have to go across this type of profile for the energy. So let's do it. I'm going to do it by brute force. I just take, use kinetic Monte Carlo, sample maybe like 1,000 trajectories, and I'm going to see, my hope is that some of them are going to end up there, and then I take this subset and I analyze the statistics. But maybe you already see the problem here is that because A is very stable, it takes a very long time to actually see trajectories arriving there because it's very difficult to escape the well A. And so this method is very inefficient, and while I can do it for these simple problems, it doesn't scale well with the dimensionality and with the size of this, the resolution you want to have and all these things. So you need something more efficient. And the core of the problem here, just to spend a bit of time, is to explain it, I'm going to introduce a couple of concepts. The first one, first passage time, I already mentioned it. So in this context, the first passage time would be the first time you arrive in a state that belongs to B. I can define a symmetrical quantity, which was the last time I was seen in a state belonging to A. And everything in between is a transition, right? Transition path. So these are examples of transition paths, and I can use those trajectories to compute the time it took for each of them to go from A to B, and this is the transition path time. So let's look a bit at their distributions. This would be the distribution of the first passage time, and this would be the distribution of the transition path time. And I don't know if you noticed something, but the thing to notice here is that this one is 10,000 times smaller than this. So this relates to what I was presenting in the beginning, is that you have this inequality, and it's very annoying because if I take back my example, by definition, I have to run the simulation for the whole first passage time. This is when I end up in the states that interest me, so I cannot escape it. So let's say it takes a year to run these simulations. So I'm spending a lot of time, and from these simulations, I'm only interested in the transition, and the transition took 1 hour of computational time to run. So it's a huge waste of time.
So you started by having W alpha beta and W beta alpha. Did you turn one of them to 0? Why do things only go in one direction? Why does A always go to B and never B to A?
I started all the trajectories from A.
Yeah, but why can't it go back once it starts
they do go back,
okay.
and that's why it takes a while before some of them actually, they spend their time basically trying to leave and coming back, and then most of them always come back, but a few of them manage to escape and eventually make it to B. Yeah.
But your definition of, can you go one more further? So TPT, so I think you're assuming that it only went from B to A and didn't go back, or I just don't understand your definition of TPT.
Well, the first passage time, so here it's the first time it's in B, so by definition the particle made it to B, and then the LPT, last passage time, would be the symmetrical concept where the particle escaped A and never returned. So
never returned. Okay.
Yeah, in the simulation I'm looking at.
Okay. Okay. Thanks. Okay.
Okay, so this is the Kramer times again, and this thing here is again basically the TPT I talk about, and again you see this inequality. Okay, yeah, I put this slide, but it's more context, but it's not strictly necessary. There have been some working on the distribution of this quantity. It's not an obvious distribution, but it's been worked out, at least in a simple case. If you consider again a general non-overdamped Langevin dynamics. So here the variance of this noise will be slightly different from the overdamped case I showed before. But you can analyze what is, basically what I was showing before, the motion of this particle in this double well potential. And you're looking basically at the particle overcoming a parabolic barrier. And it's been worked out, and this is the paper in question, and they show that the distribution of this transition pastime has this distribution, which is sort of complicated, but it has basically two parameters, which is the energy, the barrier height, so this thing here, and then the curvature, which is the curvature of the top here. So you have two parameters, and so from the simulation I showed, you can see that they fit pretty well. This distribution, which is the theory is this fit here is in orange, and those are the points. So the same distribution I was showing previously. Yes. Okay.
Is this the same eta as here?
The same?
Eta? The same, I don't know what letter you call it. The variance.
Eta. Yeah, the variance of this one is not the same variance as the
one you had before.
Yeah, it will be QBT square over D. Okay, so how do we solve it in the end? Well, to do this, we use some what I call Markovian magic. So basically, you can show, what is the real quantity of interest? What I'm really interested in is not only the probability to be in a certain state, alpha, given the initial state, which is the usual probability that you compute in master equations, but actually I also want to end up in a final state, alpha f, given that I'm in my current state alpha. So the probability I want to define is this R, which is the probability to be in the current state alpha time t, given that I started somewhere and that I end somewhere. If you take the derivative of this, you can work out that this actually satisfies also a master equation. And this master equation is related to the one which was, from the process I've shown before. It depends on exactly the same parameters through this transformation. So the rates in these new master equations are proportional to the original transition rates, but now they're multiplied by something which depends on time. And this something which depends on time enforces that when you approach the final time, you will actually transition to states which are close to the final state. I discovered along the way that this has a name in the mathematical literature. This is called actually H-dope transform. It's a Dutch physicist, so H-dope transform. what's important about it is this is sort of an effective process. It's an effective process which is conditioned to the end state, but very interestingly, it preserves the path measure. So before I said preserve the statistics, which is true, but mathematically you can show that it preserves the path measure of the original process I was studying before. And with that, you can now generate trajectory which connects the initial state to the final state. Revisiting my example, this is now how the simulation would look like. So I started in A, and I've enforced that at a certain time TF, all of them must end up in a given state in that well B. And you can see now that the trajectories are leaving A and making it to stop there, and then ending up in the final well B. Maybe, I'm not sure you could remember, but maybe something to remember that this T is now much smaller than the one it was before, so it's much more efficient to run this simulation. And why is it much smaller is because now, while before I had to run the simulations for a duration of the first passage time, now I have to run it only for this, the typical transition pass time, which is order of magnitude smaller. And so this results in a method which is more efficient and also focus on the paths of interest to you. So this is the main result I wanted to present, and then I'll move on to some possible application of this method. So as I said, our group is embedded in a biomedical research institute, so we decided to apply it to something which is very important to them, and in particular to investigate self-aid choice. I'm going to explain what I mean by that, but it's to give you a perspective. It's generally in the context of cell development. So maybe this would be a medieval way to investigate self-aid choice, right? But now we have a lot of other things, and the rage now, if you go in biological seminars, is those single-cell RNA UMAPs, and so this is supposed to represent the development, cell development. So this was generated in my institute to represent the development of the zebrafish from the egg cell all the way for the first few hours of development. I'm going to, in a few slides, give you more explanations, but maybe right now, just to give you some context, this is a UMAP, so it's a dimensional projection technique. The dots represent single cells, and the proximity on this space represents similarity in the genetic expression profiles. So basically, dots that are close mean that cells that express more or less the same genes, right? And then some biologists came in and annotated them with some color, and they say, okay, this one I recognize this cell, this gene, I know it's this cell type. And this one I know I've seen this pattern before, I know it's this cell type. And so when they annotate it, you get this colored clouds, and you see that it makes sense because cell of the cell types are close in that space because they have similar genetic expression profiles. And then the idea is that you put those cells in that space during development, and you see how they move there, and what do they become from undifferentiated cell types to more differentiated tissues. This is data, yeah. Okay, so back to more simple graphs. If I try to maybe rationalize what I've seen, I could define a network, right, where each node would be a cell, a single cell, right? And I'm going to call it now a microstate. Okay. And then you have to imagine that those cells, they have been sampled at a given time, but they are moving, right, in this cell-fate space. And maybe if you had done the sampling a bit later, they would have become a slightly different cell, right? And so I'm getting those different points, and those points represent single cells, but I'm calling them cell state or microstates. And then, cell could transition to become something which is a little bit more differentiated and with a certain transition rate. So this is another data set where, once you have this defined and, maybe you have a matrix which is with your space, you can compute all sorts of quantities. So for example, here I could compute the mean velocity emanating from a given microstate. So
sorry, I got confused. You have a three-dimensional design. The 3 dimensions is space, or is 3 dimensions something else?
It's not space. Yeah.
So what are the 3 axes?
It's they're the 3 first components of a UMAP decomposition. So I'll explain it more rigorously later, but yeah, they're not space. They are like some sort of principal components of dimensionally reduced coordinate system. Okay, so this here, like for example, will be the velocity. And so this is a 2D UMAP this time, but if I've plotted all of them with the data we had, and you see that you have a field which is not really trivial. And if you look at this, it seems like, for example, this region, the arrow points inward. So maybe here you would get trapped. And here, all the arrows seems to point toward the right. So it seems like in that portion of the space, you would move directionally, for example. And the question you people ask is, how do you go from one undifferentiated cell type, like a stem cell, to a different one, and how do you go to it? So for example, you just seen the velocity field, and I could ask, how do you go from this type of cell state to these ones? You know, I could take a triplet of racers to answer the question, and maybe the smart one will go just and take the shortest path, right? And then the other one is more like the explorer type, and we start to make all sort of convoluted loops before finally ending up in the right state. But which one is the most efficient? There is not really an answer for this, actually. And it depends on a lot of things. I mean, and this question raised different questions. For example, what are the competing differentiation routes? Maybe this one is the most likely, but maybe those ones are actually really close in terms of ranking of probability to do. Actually, maybe this one is the shortest one, but maybe it has a lot of energy barrier, so maybe actually the yellow one is the fastest. So you also have to resolve time. And yes. Okay. So now to give you a bit more information about these experiments. So they are called single-cell RNA-seq experiment. So sequencing because you sequence the transcriptome, so you get an idea of what genes are expressed, and single-cell because it's single-cell level. So without going into the detail of the experiments, which I myself not very expert about, but what I know is that what they give arrays like this, where you have like n rows, which represent single cells. You have p columns, which represent p different genes. And therefore, each entry in this table represents the number of transcripts that was detected in a given cell for a specific gene. And those are huge tables. They are very big. And so this is very high-dimensional data. And so people, to try to make sense of them, have come up with dimension reduction technique. You might have heard of TSNE. And another one, popular one, is this UMAP, which try to reduce, to identify, like in principal component analysis, a coordinate system which, by order of importance, so one of the coordinates is the most relevant, and then the next one, and the next one, and so forth and so on. And so this is, for example, a 2D UMAP. Each dot here represents a single cell. Proximity, again, represents the similarity in those genetic expression vectors. Colors is the annotation by the biologist. And this is already very informative, but people have come up with more metrics. And another type of things they've started to give us is what they call the RNA velocity. So the RNA velocity is basically representing the time derivative of this genetic expression vector. Right? So it is supposed to be proportional to the derivative of the RNA transcript. And there is a method. I mean, I can talk about it later if you want, but it's a very smart way, using the data you have to estimate those velocities. So when you use information and you overlay on that, you get things like this, where the field lines represent the velocity fields of this, RNA velocities, and it gives you some sense of directionality. So while before, it wasn't very clear what's the motion in cell-fate space. Now it's become clearer, right? It seems like cells are starting from this NMP states, which is actually one of the main precursors in zebrafish. And then from there, they can become all sorts of differentiated tissues, like a neural tube or epiderm or muscle, you know, and it gives you a sense also at what they cross to get there. So this has
drawing in the middle. This drawing in the middle that you have is just a bunch of tables like that in different times. Is that correct?
Yeah, it's yes, exactly. Yeah, they have a bunch of tables like this for different time points.
Yes.
And then they are usually they're even aggregating them together because, you know, from the idea that although they have been generated at different time points, the cell types are very different at time 0 or at other times, so they would be separated in that space.
Right. And just out of curiosity, what is the frequency that these things are sampled?
2-4 hours, something like this.
2 or 4 hours. Okay.
Okay. So this is very informative, but something that you don't see with that is you don't have a clear idea of how long it takes to go from there to there. I mean, it's not really. And sometimes you have bifurcations. For example, if you follow this red path, it seems like you reach there. You could go left or you could go right. And so again, you can look at it, but qualitatively have an idea. But if you want quantitative understanding, it was sort of missing. Okay, and so this is where we decided to apply our methods. And so to be perfectly clear, what I've called before, you know, single cell, as I said, are abstracted as microstates. So this is, you have to view it as a continuum of states, and the points would represent like a discrete sampling, the experimental sampling of it. And then there are methods which have been published, which use those RNA velocities to compute transition rates. So, you know, the time units can be the subject of controversy, but at least there are some methods which produce transition rates. So once I have this, I have basically all the ingredients necessary to apply the Markov bridges method and to sample paths between any two arbitrary pair of points on this landscape.
Right, from the one table to the other, you have a transition.
You mean, yeah, okay, so if I understand correctly, you could actually start from there and then follow the velocities, that's what you're saying? Yeah. And then arrive to different points. So you could get, you're right, but then you would get probably the most likely trajectories because you follow just the velocity, so you only have one. And then, you get the time for this one, not for the others.
Do you have to make a hypothesis that the transition rates, this alpha, W alpha beta, are time independent? Is that a hypothesis that you have to make?
Well, the one in our application, at least, they were all, you know, all those time points, they were all like bundled together. And so you get, you know, maybe if you were only plotting the table at time 0, you would get just this yellow and orange one.
Yes.
And then if you would plot the one at a later time, you would get only the green and the blue and this light blue and the purple. And then if you do at a later time, you would get only this one and the leaves, basically. But if you put all of them together, you get the whole picture, and the transition rates are computed from the whole concatenation of all this data.
Okay, thanks.
So I don't know if it's related or unrelated, but how do you define the distance between two different points? So you have some axis, you didn't tell us what it is yet. So I don't know how you decide.
Well, so the UMAP is...
how do I tell if 2 points are very close or very far?
So ideally, you should, the best plot probably would be a p-dimensional plot, right, where each axis would be a given gene, and then the proximity in that space would tell you that the cell at the same, very similar genetic expression profile, at least the same number of transcripts, you know. Okay. And then I'm not really, it's not really my expertise, but you know, data scientists have this way to use clever PCAs, and so to figure out like a, you know, a system of coordinates in which you can only take the first few ones and to make sense of your data, and so they reduce the dimension of the data set. And so in PCA, maybe you would retain the first 3 or the first 2, and then you would get, if you plot the points into that system of coordinates, you would get this. So
if I change my definition, I mean, it seems like there are arbitrary ways to change it, but is the result the same? I mean, I'm asking, is your result invariant under different choices of these axes, or?
I'm not sure. I don't think there is any, proof or any rigorousness in which people apply the UMAP. There is even some stochasticity in the algorithm for the UMAP, but the deal is you trust that what you get should be, a better representation which preserves the proximity between the genetic vectors.
Okay.
Yes. Okay. So in the application I'm showing now, we chose a data set of pancreatic cells in the mouse, and in the pancreas you have Langerhans silence, which are where endocrine cells are, and you have 2 types: the beta cell, which produce the insulin, and the alpha cell type, which produce the glucagon, which is has the opposite effect of the insulin. And those cells are basically, when they are during development, they come from pre-endocrine cell types and from those ductal cell types. And so if you look at this, you do the same thing. You take tissues of pancreatic cells in the mouse, you sequence them, you compute the UMAP, and you plot them, you get this, right? And so you have different cell types, and I'm telling you, this area corresponds to the ductal cells, and this color in orange corresponds to the beta cell types, and this in blue corresponds to the alpha cell types. And I can ask, well, how do the cells differentiate from this ductal cell type here to either the beta cell types or the alpha cell types? And this is exactly like a good application for our method, right? So here are some examples of trajectories that are the result of this. And qualitatively, you can see maybe already that whether you differentiate to beta cell types or alpha cell types, a lot of trajectory remains stuck in this ball for quite a while. So it seems very hard, once you reach this collection of states, to escape it. However, once you escape it, they seem to move quite directionally toward their respective end state. They don't really come back, right? So you can use, make more quantitative what I just said in words. So if I focus, for example, into the bottom movie, which was the differentiation into the alpha cell types, this would be a time lapse. And so what I said earlier about the fact that the cell remains stuck in one region, you could just formalize it by defining the waiting time. So you have these colors, so red, pink, brown, which correspond to different cell types. I can define a waiting times, how long it takes for a trajectory, in the red state before it leaves and doesn't return, right? So the waiting time would be the time until it leaves that state forever in that simulation. Well, you can see that those two are the most, are the one with the largest waiting times, and so these states, they behave as traffic bottlenecks. Similarly, what I said with some cells moving quite directionally, well, you can just look at what is their velocity in that space, and average its direction, and while here it is roughly 0 because the cells are stuck and are going in all sorts of directions. Once they are in this purple, brown, or gray cell types, they tend to really move toward, conserve their directions. So those cell states behave more like freeways in a sense. Okay. And something else you can do, which actually I think is one of the main advantage of this method over, yes.
Sorry. so you have a snapshot. These are simulations or data again? Sorry about this.
So the dots are, are data from experiments, and then the trajectories, which are a bit small, they are the simulations.
And they're different snapshots.
Yes.
So the question is, why the shape does not change?
Well, because the shape is, I'm assuming again that I combine all those time points together, and this is cell-fate space. And I understand that they start somewhere at the beginning, in the early development, and then they transition toward a different region of that cell-fate space. And so if you were doing the sampling, maybe at the beginning of the experiments, you would get those points, and later on, you would get those points, but I'm just grouping them together.
The shape do change, but you're just grouping everything together.
Yes.
Okay.
Okay, and so the last thing is what I was mentioning earlier is what about bifurcations, right? And the good thing with this method, so here I only plot the fraction of the trajectories once they leave this brown state for good until they reach the blue alpha cell type for the first time. And what you can see is that we now see the vast majority of the trajectories use this lower branch, and then the remainder is roughly equally split between the other top branches. And this is not something you could really see from the RNA velocity and these static pictures. So I think this is something really, a really valuable insight got from this method. And you can compute the statistic of these different branches, but you can also therefore compute the kinetic rates to go from this one to the blue state using different routes. And that's also something else you can compute. Okay, so here are my conclusions. And we have a preprint about this. It's still in the review process, but so if you have any suggestions, it's a good time because we can incorporate them. And also, if you're interested in trying to play and generate Markov bridges, I did a little Python package, and so same thing. If you have any suggestion, it would be great. And yeah, with that, any questions?
So, about your method, is it reminiscent of stochastic resetting in the sense that you change the original microscopic trajectories in a controlled way, and then you correct at the end to find the actual set of probabilities in the steady state?
Not exactly, because I think stochastic resetting, you leverage the Markov property, right? So you go to somewhere intermediate, and then you break the problem into two, basically. You go first half, and then you look at the second half, right? Or make more stops if you need. But this is like the correction terms is there always, and the correction terms is time dependent, and it is also this is computed through. Because we know from the system you can compute exactly the probability to end in the final state given your current state. So it is more expensive to compute. But because you have to run smaller simulations, overall it's faster.
Efficient. Yeah. Thank you.
Yeah, thanks. So I was wondering that you put the formula for the Kramer's time, and there was the expected time that was logarithmically, so that there is like a kind of physical intuition about the logarithmic, and I am asking that because perhaps really with the other question about the Levy flights, if you have a Levy process, so the paths are like more longer, so perhaps that time scale for the jumps also decreases a lot. So I'm like willing to know if that logarithmic is kind of universal or it's only for Gaussian process, or what is?
Well, it was computed for a parabolic barrier, so okay. There you have your Gaussianity, in fact. So I think, yeah, it is something which is, in principle, only valid for, when you are close to the barrier, right? You can more or less assume it's Gaussian. But funnily, it seems to hold quite well when you extend it to barriers which are not exactly parabolic and where you deviate from Gaussianity. I don't have a really good answer to that. Yeah.
Okay. Okay. Thanks.
Other questions?
So I have a question. Just to try to understand
Yeah.
what you have done. So you have a model for these transition paths, right?
Yes.
So what are the parameters that define your model? Are the jump probabilities, this W, alpha, beta?
Yes, that's the parameter.
And that you can measure from
Yeah.
the experiments. And then I'm just imagining. So suppose you take the experiment and you do these measurements for 2 days. But then you take the first day and then you calibrate the parameters of your model. Then you can predict what's going to happen the second day and compare with the results. Is that does it make sense, this logic?
Yeah. Yes, well, I think maybe you could do what you're, I mean, that would be great. But I think maybe you could do that if for example, when you were sampling at the early times, you were getting a little bit of everything, right? Because then you have the transition rates from this region, and also from these regions toward this region. But when you do it experimentally, you start by having just this, and then you get, maybe just this, and then just this, and so you don't even have the states that would correspond to the more differentiated ones.
So what do you predict then? How do you know your model is correct?
Yeah. Well, for the application to, I don't think, well, for the application to self-aid, I think it's still ongoing, right? Because there are different sources of bias in the experimental data that we would need to show that the method doesn't really, exacerbate or amplify. For example, one of the experimental bias is you don't get all cell types, you don't sample all cell types in the same way. Some of them have a shorter lifespan, for example, or proliferate faster than others, and so some cell types may be overrepresented. So basically, the density of points is not really uniform, and so then you get transition rates between microstates, and in some regions, those pair of points are much smaller, and so something, for example, we would need to show now that in principle it works well when the distribution is homogeneous, but when you have this inhomogeneity, we need to show that the method doesn't give you something which doesn't make sense. Yeah.
Okay.
Yeah.
And just a provocative
Yes.
question now. Machine learning is everywhere. Can you use machine learning to learn how this process evolve, or are people doing this? Is this a reasonable thing to do?
Yeah. Well, I didn't do it. I know, there is, I know in our institute there is a new team being created, which is going to be called the virtual cell. And so virtual cell. So they're interested in this type of, they want to be able to use basically machine learning to predict those features. I don't have a really good answer. I don't know. Is it going to be better than physically informed methods? I think at some point you need to have some physical or biological input of some sorts, but I guess we'll see.
Thanks.
I can ask the same kind of question like him. Suppose it works, your model works, or suppose it doesn't work. What have you learned? I mean, I'm just trying to understand. What's your model? Your model is a new parameter you include, or your model is a new way of the thing moving? Or?
Well, I think what we learn, I think, is if you ask me how you go from there to here, I can tell you new things that this couldn't really tell you. I can tell you, what are the alternative paths around the most dominant one that you would have by integrating the velocity field. So, and in particular, you can identify rare trajectories. Maybe a possible way is actually to maybe do a little detour and then go back here. And
so your model samples more paths. Is that what you're saying? Than the
you get all the paths, and you also the statistic is preserved, and so doing that you can put numbers like the probability of using a certain route versus another, and those are things I think. But I think it's interesting to have this calculation available, and I don't think it was available before. Yeah.
Thanks. Any other questions? Okay, let's thank Yannick.